The DNA repair systems that we talked about, like base excision repair, nucleotide excision repair, they rely on the template strand of the DNA. But what happens when there is a double strand break? Like this is easy to repair because there is a template strand and the polymerase can fill in this gap. But if there is a missing part in both of the strands, then it creates a problem. Actually, this is a more serious problem. And if you have watched the CRISPR video that I published last week, the CRISPR-Cas9 system uses the double-strand repair pathway. So it creates a double-strand repair and then double-strand break, and then the cell repairs it. And while repairing, it introduces some mutations or deletion of the gene. Why deletion? Because we have first mechanism for double-strand breaks is NHEJ, which is non-homologous and joining. And joining because if there is a double-strand break, two proteins come in here. This is called proteins. Come and bind to the opponents, and they process a little bit. So they cut a little bit from here so that the ends will be how to say, blunt ends, as I say, because if there is a sugar is missing on one end and there is a phosphate missing on the other end, then you cannot join them. There should be a perfect missing nucleotide so that the new nucleotide can be added. Proteins first make these parts, achieve these parts, and then it connects the two strands. And there is this piece would be missing. So this is not an ideal repair, and it may sound a little bit silly for you because this is not a repair, it just patched up two strands that were broken. But if the double strands breaks are left unrepaired, they cause more problems such as like chromosome instability or chromosomal damage, which is a huge problem for the cell. And if there is a chromosomal damage, the cell should die immediately, like goes into apoptosis. So patching these two together is a compromising but acceptable solution. It is non-homologous because it doesn't use any homologous strand, and end joining because it joins these two ends. And then we have homologous repair. Homologous repair uses the fact that each of our cells have two copies of the same chromosomes. So one is coming from the one is paternal and one is maternal. And if there is a gene in here, there should be a copy in here as well. There might be some differences like SNPs or allele differences, but it doesn't cause too much problem. So it has a homologous strand. And in this situation, the break in this one is repaired by using this one as a template. Why doesn't it use it on the NA, for example, instead of this, why doesn't the cell use HR all the time? Because for HR to happen, the cell must be dividing so that these two chromosomes come together. Because in the normal life cycle of the cell, the chromosomes are like this. Whenever they become condensed and taken together, then the homologous recombination can take place. And also, as you may have heard probably, crossover also happens during this stage, and crossover happens through homologous recombination as well. So homologous recombination is both repair system and also meiosis, it is used in the meiosis to create variation for the offsprings. How does it use it? The chromosomes are in the condensed position, so they have to be opened up first of all, and then the repair can continue. So we have this DNA, and then we have this broken chromosome. After these are both opened up, then some proteins bind in here and carry them to their matching part. So the situation becomes something like this. With the help of the proteins, they search for the matching site, and for this as well, like this. Now this can repair itself, and this can repair itself as well. Then what happens is, for the repaired part is this. Sometimes they might not perfect, they don't have to perfectly synthesize. For example, they may lose these parts, and then they can do the replication by looking at the other strand, because now it doesn't require a homologous chromosome. This is quite easy to repair, so it will need only DNA polymerase and a ligase. The proteins taking part in this situation we will see in a minute, and this can happen only during the S phase and G2 phase, it says, not in the G1 phase, for example. All other DNA repair systems can take place any other time in the cell. The one thing we have to be careful about is the DNA synthesis has to continue from 5' to 3'. So this one, because it is continued like this, should be 5', and because this is continued like this, this should be 5'. So this is 3, and also on the other side, this is matching with this one, so it is 3', 5', and like this. And the direction of the synthesis is like this and like this. So this is important because this is the 5' to 3', and its complementary strand is this one, and this has a complementary strand in here. So it might become important whenever you think about how it will be repaired. So in the CRISPR-Cas9 experiments, for example, you have to think about this very carefully, because if you want to change something or delete something, then the directions will be important. Homologous recombination is called homologous recombination because it's actually the same as the homologous repair, but it starts without a DNA damage. Why it starts? Because to create the crossover products, and it only happens during the meiosis and the first stage of the meiosis, and it is used in the creation of the gametes and the variation of the offsprings. And this is a very conserved mechanism, and it is even present in bacteria and yeast. The homologous recombination is the same thing with the homologous repair, but in this situation, homologous repair mechanism cuts the DNA itself. So it cuts this and then it starts. In homologous repair, the DNA was already damaged, double strand. So there is a nuclease digest in here, and this one is called a strand exchange. Strand exchange because the two chromosomes exchange their strands. And at the end, as always, there is a polymerase and a ligase. Before going into this, there is a terminology here. So this is a DNA duplex, a normal DNA duplex, and this is called hybridization. When it is a hybridization, when this strand comes and binds to it, like in this situation, it comes and binds to it, it's called a hybridization. And in homologous recombination, there must be a perfect match. But it's not the case all the time. And whenever there is a strand exchange like this, so this is the paternal and maternal chromosomes, and the strand exchange has happened. This is called a heteroduplex. Heteroduplex because the two strands are from the different molecules. And the heteroduplexes, there are mismatches, like this one. And in the homologous recombination, it omits those differences and then repairs later if it is necessary. So out of the 20 or 50 nucleotides, if the 45 is matching, then recombination starts. And this is also another way of introducing variation. Now there are a group of proteins associated with this system, and it is called the RAD51 in human and REC-A in E. coli. RAD51 in eukaryotes, sorry. And these are the most important proteins for this homologous recombination event, because they start the strand invasion, strand exchange. Sometimes it's called strand invasion. They start the strand exchange process, and they require ATP for doing so, because you will take one strand and take it to the other part. Obviously, DNA repair systems use ATP as well. They attach this one and facilitate the exchange between the strands. Now, what it says is it searches for a similarity, and when it finds a similarity, and if it's a perfect hybrid, if it's a perfect match, then ATP is utilized to make the hybridization. So before that, it doesn't use any ATP. It says almost perfect. So to prevent it going into this one, they destabilize the strand. So the first thing this strand is going to make a complement is this one, yeah, its own molecule, but the proteins carry it out so that the strand exchange is successful. So this is a type of like a forced event onto the chromosomes. Then we have another critical definition, which is loss of heterozygosity. Loss of heterozygosity you will see in the cancer subjects, but before that we have these homologous chromosomes, and these will have the same genes. For example, if this part is coding for the gene A, then the sequence will be the same, TAGCT. The sequence will be same because it's the same protein, same gene, but sometimes there are some changes in here. For example, this might become an A, and this is called SNP, small nucleotide polymorphisms. They are not called mutations most of the time, because most of the time they do not cause a difference. If you sequence the same gene for 1,000 people, you will find lots of SNPs. But as a more technical term, the SNP is a change in the DNA, a mutation, for example, a change in the DNA, which has a less than 1% occurrence in a society, in a group of people. So if the A is changed in only 0.5% of all the people in Turkey, then it is called a SNP. But sometimes they cause mutations. So if this change, like this A, changes the protein, changes the function, affects the function of the protein, then it is called a mutation. For example, if this gene A is P53, which is a tumor suppressor gene, so it prevents cells from going into cancer, and this one is mutated, and this one is wild type. So this is producing the protein, but this is producing nothing. But because P53 is required at a very low levels in a cell, if one of the copies, one of the chromosomes, homologous chromosomes, produce the protein, then the cell is again has enough P53 for its life. Then if during the homologous recombination or during a repair, homologous repair, this is synthesized by looking at this and converts itself into an A, then this will stop producing the protein as well, and then there will be no more P53 in the cell, and this is will be a cancer cell most probably. This is called the loss of heterozygosity, because it had, and in the beginning the cell was heterozygous for this locus, but then it lost it, so it is called the loss of heterozygosity. Lost allele for an important protein, for example. Now P53 is a very important protein or gene, and like 50% I think cancers have mutated P53. So if a cell has a mutated P53, then it goes into cancer. Why? Because in the beginning of G1, P53 sits in here, and if there's something wrong in the cell, it forces cell to go into a G0. But the P53 is missing, then the cell continues dividing without bypassing the checkpoints in the cell cycle. And the other thing, you may have heard Brca proteins. You may have heard that Angelina Jolie has Brca mutations, and because her mother had died from the breast cancer, and she had mastectomy because she had the same mutations. Why? It was a wise choice, because Brca proteins are accessory proteins of the RAD51. RAD51 accessory proteins, Brca 1 and 2, there are two of them. In the homologous recombination or homologous repair, they stabilize the RAD51 protein so that the strand invasion can happen, and it processes the ends. If the ends are not processed, then the cell goes into any non-homologous end joiner. So they are the critical proteins for the double-strand breaks. So what happens in a BRCA1 or 2 mutated cell, then whenever there is a double-strand break, it won't be repaired, and then chromosomal instability will happen, and that cell will go into cancerous pathway quite easily. In the homologous recombination during meiosis, there are other proteins like SPO11 or MRE11, and there is a Holliday junction forming. If you remember, after the strand invasion, they dissolve, and each chromosome becomes itself again, not entangled with the other one. But during meiosis, they stay like that, and then the enzymes cut from different directions, and wherever they cut that, they create different results for the cells. For example, this would be the we have this one and this one. There will be a break in here, for example, and then we will have like this. So if it's a homologous repair, then the result is like this chromosome is unchanged, and this chromosome is changed a little bit. It has a sequence coming from this one. But if in a Holliday junction and homologous recombination, how it cuts from the strand invasion creates like what you saw as a crossover product. What is a crossover product? Like this. So it seems like these two have exchanged pieces. Like crossover is called as a exchange of the chromosomal parts in the cell, but it's actually not an exchange. It wouldn't make sense to cut this and paste it in here. Instead, they entangle each other, and then they synthesize by looking at each other, and because of the cutting, they change their parts. So it is not possible to for me to describe this on the 2-dimension here. So I will link some videos below so that you may imagine how it's happening. Some of the videos are very simple, so it's better to understand what's going on, and some of them are a little bit more complicated. And there are also other proteins, a lot of proteins involved, and this is a very specialized